All of you? So we have now digestion of food done by the pancreatic juice. The pancreatic juice has in it many inactive enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidase, amylase, lipase and nuclease, of which trypsinogen, chymotrypsinogen and procarboxypeptidase help in digestion of proteins. Amylase helps in digestion of carbohydrates. Lipase helps in digestion of fats. And nucleases help in digestion of DNA and RNA, which are the genetic materials found in the nucleus of the cell. So, sir, pancreatic juice is practically breaking everything. It is breaking down proteins, it is breaking down sugar, it is breaking down fats, it is breaking down nucleic materials, and उसमें how does digestion take place? So let's understand how does pancreatic juice work. Pancreatic juice has in it amylase. Amylase acts on starch to form disaccharide maltose. Hey, ये सुना हुआ है. If you remember, tyrene had broken down polysaccharide starch into disaccharides called as maltoses. Similarly, pancreatic juice also has in it amylase, which breaks down polysaccharide starch into disaccharides, which are called as maltoses. Difference being, the salivary amylase acted at a acidic pH of 6.8. The pancreatic amylase will break down starch in a alkaline media. Pancreatic juice, if you remember last lecture we did, has a pH of 8.8. So at an alkaline pH, starch will become maltose with the help of pancreatic amylase. So sir, 30% of the starch was digested by salivary amylase or tyrene, and 70% of the starch is broken down by pancreatic amylase and starch is digested into maltose. Then you have lipases. Lipases are activated by bile salts. अभी मैंने बताया था, bile salts, sodium glycocolate and sodium taurocolate are produced by liver, but they enter the intestine, and when pancreatic juice enters the intestine, pancreatic juice has in it lipase. Lipase is inactive. Bile salts, sodium glycocolate and sodium taurocolate will activate the lipase, and then pancreatic lipase will break down lipids or fats into smallest possible particles called as fatty acids and glycerol. So pancreatic lipase breaks down fats into fatty acid and glycerol. If you remember, what did bile salt break down? Fats into smaller particles called micelles. And then what does pancreatic lipase do? Break down micelles into small particles called as fatty acids and glycerol. And then we have nucleases. Nucleases in the pancreatic juice acts on nucleic acid to form nucleotides and nucleosides. तो सर आपके DNA के अंदर आपका सेल होता है, सेल के अंदर होता है न्यूक्लियस. न्यूक्लियस हैज इन इट DNA, and DNA consists of nitrogen bases called as adenine, thymine, and you have guanine and cytosine. And adenine, thymine, guanine, cytosine are nitrogen bases. Attached to the nitrogen bases are found sugars. So what is nitrogen base attached to a sugar? And ये है sugar, and sugars are attached to phosphate. Sugars are attached to phosphate. And we have one nitrogen base and one sugar is called as a nucleoside. A nitrogen base and the sugar is called as a nucleoside, and the nucleoside plus the phosphate attached to it is called as a nucleotide. So sir, what is nitrogen and sugar called as a nucleoside? What is nucleoside plus the phosphate attached to it called as a nucleotide? So what is nitrogen, sugar, phosphate, nucleotide? 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 So sir, what is the DNA made up of? Polynucleotides. Polynucleotide. So something very nice I'll tell you. One DNA has in it 3.3 billion such pairs of polynucleotides. कितने sir? 3.3 billion pairs of polynucleotides are present. That is the amount of nucleotides present in one human DNA, which is found in the nucleus of the cell. And sir, you need new nitrogen bases, new sugars, new phosphates for one cell to become two cells. तो सर जब आगे जाके सेल डिवीजन होता है, हमारे बॉडी में ग्रोथ हो रहा है, सेल डिवीजन हो रहा है, तो डीएनए रेप्लिकेट होना चाहिए. तो एक सेल का जब दो सेल बनेगा, जब जाइगोट का दो ब्लास्टोमियर बनेगा, then the DNA replicates and chromosome number remains the same. But for DNA to replicate, you need the basic substances which form DNA. Which are the basic substances? Nitrogen bases, adenine, thymine, guanine, cytosine, sugars and phosphates. And DNAs are having in them all of these arranged in the form of nucleotides. तो सर हम लोग जब खाना खाते हैं, खाने में स्टार्च आता है, कार्बोहाइड्रेट्स आते हैं, प्रोटीन्स आते हैं, फैट्स आते हैं, और हर एक सेल के अंदर न्यूक्लियस होता है, हर एक न्यूक्लियस के अंदर डीएनए होता है. एंड सबसे पहले pancreatic juice has in it nucleases which will act on these DNAs, and nucleases break down nucleic acid. Which are the nucleic acids? DNA and RNA. Nucleases break down nucleic acid, DNA and RNA into smaller pieces which are called as nucleotides. So entire DNA, 3.3 billion base pairs, 3.3 billion pairs of nucleotides is broken down by nucleases into individual units which are called as nucleotides. तो ये पूरा DNA का nucleotides बन जाता है. And then there are chemical enzymes called as nucleotidase, which converts nucleotides into nucleosides. So nucleotidase breaks away the phosphate. Phosphate को तोड़ देता है so what will nucleotide on losing the phosphate now become sugar and nitrogen base, which is called as nucleoside. So nucleotidase removes the phosphate and converts the nucleotide into a nucleoside. And then you have nucleosidase, which breaks down nucleoside into simple nitrogen bases. Nucleosidase breaks down the bond between sugar and nitrogen base, and it forms sugar, it forms nitrogen bases, and individual nitrogen bases and sugars are formed. And what does the intestine now have in it? Nitrogen bases, adenine, thymine, guanine, cytosine. Along with it there are sugars, and along with it there are phosphates. So the entire DNA or RNA has been broken down. Once again, what will nucleases do? Break down DNA and RNA into individual units which are called as nucleotides. And what will nucleotidase do? Remove the phosphate and convert it into a nucleoside. What will a nucleosidase do? Break down the bond between nitrogen base and sugar to form nitrogen bases, adenine, thymine, guanine, cytosine, sugars and phosphate. इस तरह से डीएनए को पूरी तरह डिसॉल्व किया जाता है with the help of pancreatic juice, which is called as nucleases. And pancreatic juice also is rich in protein breaking foods. Protein breaking foods पैंक्रियाटिक जूस के अंदर होता है इनएक्टिव प्रोटीन ब्रेकिंग जूस, इनएक्टिव trypsinogen and chymotrypsinogen. So what does pancreatic juice have in it? Inactive trypsinogen and chymotrypsinogen. And trypsinogen and chymotrypsinogen are inactive. Along with pancreatic juice, in the intestine is found intestinal juice. Intestinal juice में होता है एक enzyme called as enterokinase. So what does intestinal juice have in it? An enzyme called as enterokinase. Enterokinase acts on trypsinogen and converts inactive trypsinogen into active form, which is called as trypsin. Trypsinogen becomes trypsin. Who activates it, sir? Enterokinase from the intestinal juice converts trypsinogen into trypsin. And then trypsin will break down proteoses and peptones into polypeptides. I hope you remember, stomach में pepsin had broken proteins into smaller chains called as proteoses and peptones. And now trypsinogen becomes trypsin, and trypsin breaks down proteoses and peptones into still smaller chains which are called as polypeptides. Polypeptides are chains of amino acids. So what was the whole protein, sir? A big chain of amino acids. What did pepsin break it into? Smaller chains which are called as proteoses and peptones. And who will further break it, sir? Trypsin. And what will trypsin break down? Proteoses and peptones into still smaller chains which are called as polypeptides. So trypsin breaks down proteoses and peptones into polypeptides. And trypsin also acts on another pancreatic juice which is called as chymotrypsinogen. And chymotrypsinogen, which is inactive, now becomes an active chemical called as chymotrypsin. So trypsin दो चीज करता है. Trypsin breaks down peptones and proteoses into polypeptides, and trypsin also converts inactive chymotrypsinogen into active chymotrypsin. And chymotrypsin के साथ, you remember there was a chemical called as procarboxypeptidase. Procarboxypeptidase is acted upon by trypsin, and procarboxypeptidase becomes carboxypeptide. Procarboxypeptidase now becomes carboxypeptidase. ये भी आपके बुक्स में दिया नहीं है. Please write this down that trypsin converts chymotrypsinogen into chymotrypsin and procarboxypeptidase into carboxypeptidase. And further on, sir, chymotrypsin and carboxypeptidase, act on polypeptides and break it into dipeptides. So what does chymotrypsin and carboxypeptidase do? Acts on polypeptide, which is a chain of amino acid, into two amino acid chains which are called as dipeptides. So once again, what does pepsin break? Pepsin in the stomach breaks down protein into smaller chains which are called as proteoses and peptones. What will trypsin do? Break down proteoses and peptones into still smaller chains which are called as polypeptides. And what are chymotrypsin and carboxypeptidase breaking? Polypeptides into still small chains which are called as dipeptides. So sir, इस तरह से dipeptides बन जाते हैं. And digestion by pancreatic juices completed. Last but not the least, it will be now intestinal juice which will help in the final digestion process. So what will intestinal juice do? So let's talk about intestinal juice. Intestinal juice is called as succus entericus. What is intestinal juice called as? Succus entericus. So it has in it mucus secreted by the Brunner's glands, which makes the pH of the intestine 7.8. If you remember, Brunner's glands found in the submucosa of the duodenum have in it mucus, and mucus will act at an alkaline medium, a pH of 7.8. So pH of 7.8. याद रखना all of you, mouth has in it pH of 6.8. Stomach has in it pH of 1.8. Pancreatic juice has a pH of 8.8. And intestinal juice has a pH of 7.8. Everything ends with 0.8. 6.8 mouth, 1.8 stomach, 8.8 pancreatic juice, 7.8 intestinal juice. And along with it, do we remember in the intestine there are a lot of secretory cells found in the crypts of liver crypt. क्रिप्स ऑफ लाइबरकन जो विलाय के बीच में थे, उनके क्रिप्स ऑफ लाइबरकन में इंटेस्टिनल जूस सिक्रीट होता है कॉल्ड एज म्यूकस एंटेरिकस. इट हैज इन इट डाइसैक्राइडेज. नाम से मालूम पड़ रहा है डाइसैक्राइडेज. Ase means to break. So disaccharidases will break disaccharides, and dipeptidases will break dipeptides, and lipases will break down lipids, and nucleosidases will break down nucleosides. So once again, disaccharidases break down disaccharides like maltose, sugar. Dipeptidases break down peptides, in other words, proteins. Lipases break down lipids, and nucleosidases break down nucleosides, DNA, RNA. So, sir, intestinal juice may be sugar, protein, fats और DNA, RNA ब्रेक करने के सारे केमिकल्स होते हैं. Let's see how does this happen. Sir, सबसे पहले maltase नाम का एक disaccharide होता है which acts on maltose to form glucose. So what does maltase act on? Maltose, and maltose is a disaccharide made of two glucoses. Maltase उन दोनों को सेपरेट कर देता है. So maltase breaks down maltose into glucose and glucose. Maltase breaks down maltose into glucose plus glucose. And then we have sucrase. Sucrase will act on sucrose. Sucrose is found in sugarcane juice. तो समझो आपने गन्ने का रस पिया, उसमें सुक्रोज होता है. And intestinal juice has in it sucrase which acts on sucrose to form fructose and glucose. So what is sucrase break down sucrose into fructose and glucose. And then we have lactase acts on lactose. Milk sugar is called as lactose. Milk sugar called lactose. Lactase acts on lactose to form glucose and galactose. So lactase breaks down lactose into glucose and galactose. Sir, so sir, all your sugars have been broken down. चलो उसको रिवाइज करते हैं. पहले salivary amylase breaks down starch into disaccharide maltose. Then pancreatic amylase also breaks down starch into disaccharide maltose. And then maltase breaks down maltose into glucose and glucose. Then we have sucrase which brings down sucrose into glucose and fructose. And then we have lactase which breaks down lactose into glucose and galactose. तो ये monosaccharides बन गए. Simple absorbable monosaccharides, glucose, fructose, galactose, and they can be easily absorbed from your intestine. And sugars digest हो गए. Then we have dipeptidases which act on dipeptides and form amino acids. Dipeptidases break down dipeptide and break a dipeptide into two individual amino acids. By the way, amino acids are connected by a bond called as peptide bond. So dipeptidase breaks down the peptide bond and breaks the dipeptide by breaking peptide bond into individual amino acids. तो उससे amino acid बन जाते हैं. ये तो प्रोटीन भी डाइजेस्ट हो गया into amino acids. चलो let's revise. In the stomach, HCl acted on inactive pepsinogen and converted into active pepsin. Pepsin breaks down protein into smaller chains called proteoses and peptones. Then in the intestine, enterokinase acts on pancreatic juice and converts inactive trypsinogen and converts it into active trypsin. Trypsin breaks down proteoses and peptones into smaller chains which are called as polypeptides. Trypsin also acts on chymotrypsinogen and converts it into chymotrypsin. Procarboxypeptidase into carboxypeptidase. And chymotrypsin and carboxypeptidase break down polypeptides into dipeptides. And in the intestine, dipeptidase will break down once again, dipeptidase will break down dipeptides into amino acids. इस तरह से प्रोटीन भी पूरी तरह डाइजेस्ट हो गए. And then we have fats converted, emulsified fats. What are emulsified fats? Is also called micelles. And micelles are acted upon by lipase into fatty acids and glycerol. So you remember, pancreatic juice has in it lipase, and intestinal juice also has in it lipase. And both lipases will break down fats into basic units which are called as fatty acids and glycerol. So इस तरह से fats भी digest हो गए. What does sugars become? Glucose, fructose, galactose. What has proteins become? Amino acids. What has fats become? Fatty acid, glycerol. What has DNA, RNA become? Nitrogen bases, adenine, thymine, guanine, cytosine, sugars and phosphate. Everything has been digested, and this acidic chyme of the stomach is now converted into a liquid form called chyme. Stomach में क्या था? Chyme. It was acidic. Intestine में after digestion is over, what has it become? Chyme. So chyme has become chyme, and chyme is alkaline, and chyme has in it everything in the simple form: sugars, amino acids, fatty acids, glycerol, सब basic simple substances हो गए. So where did maximum digestion take place? Maximum breakdown takes place with the help of bile, pancreatic juice and intestinal juice in the duodenum. So maximum breakdown, where does digestion mainly take place? In the duodenum. And where does maximum absorption take place? Absorption of the digested food takes place in the jejunum and the ileum. So where did digestion mainly take place? In the duodenum. Where did absorption take place? In the jejunum and the ileum. And that is the way food has been completely digested and has got absorbed. So we have understood everything. Today's lecture, beautiful concept we have understood, the physiology of digestion. इसको बहुत अच्छी तरह पढ़ना. नीट एग्जाम में 100% एक एमसीक्यू आपको आज के लेक्चर फिजियोलॉजी ऑफ डाइजेशन से जरूर आएगा. Please study this part thoroughly, well, all of you, and ask me your doubts in the Zoom session. Work hard, guys. Take care. God bless you. Bye bye.